So in this short talk, I would like to show you what is the BabyLux project, just to put everyone on the same page to better understand the clinical results and the perspective for the future. So I will briefly start with the motivation for running this project. Then I will briefly speak about the technology involved, and then I will briefly deal with the business model that we have implemented together with our company partner, and then, present some basic results. Okay, as already shown, the motivation is related to the fact that every year, according to the World Health Organization, we have around 15 million preterm babies, and this counts up to 5 to 18% of all babies born. So this is a huge problem. Medical doctors may have several technologies to deal with this problem, in particular to monitor what is happening in the very first hours or day of life. In particular, we would like to focus on cerebral oximetry. Cerebral oximetry is a technology which uses light, in particular red and near-infrared light, to probe biological tissue in depth. The principle is quite simple. So in this spectral range, we have a very low absorption from water and blood, which are the main constituents of biological tissue. So light can penetrate deeply. And thanks to another physical phenomena, which is the scattering, light can also escape from the tissue. So we can make a non-invasive measurement of what is happening inside the brain, in this case. So this technology, this approach has been already used in the field, and several nice devices are already present. I just put here some European, Japanese, and US devices. So they are fancy, nice, expensive, probably. However, from our point of view, the point is that they have some problems. Problems in quantification of the properties of what they measure, reproducibility, and most of them, they don't give information on the complete hemodynamics, so they don't give information on blood perfusion, for example. So this was the starting point, the fact that there was an unmet need in the clinical care. So we wanted, with this project, to provide a solution, a possible solution. So the idea was to provide a precise, non-invasive, robust solution to provide cerebral oxygenation and blood flow in a non-invasive way. So the project started 3 years ago, enough already, for 40 months, and included a multidisciplinary contribution from 4 partners from European countries, Italy, Spain, Germany, and Denmark. From the point of view expertise, we had technology expert in Barcelona and in Milan for Photonics. Then we had the support of SME and company and the Research Institute for Industrial Production in Fraunhofer. More important, we had the contribution of end user. So in this case, our hospital in Copenhagen, Gorm Greisen, and in Milano, Clinica Mangiagalli, Monica Fumagalli and colleagues. And most important, we had the possibility to exploit the enthusiasm of a new company, so Photonics, and the support of Fondazione Politecnico di Milano to bring this device to reality. And just a few things about the basic physics behind. So we are using photonics, but let's say advanced photonics technique. So on one side, we use time-resolved near-infrared spectroscopy, shortly TRS, which means basically cerebral oximetry with pulsed light, with pulsed picosecond light, so a very sensitive, very high time resolution technique, which has the ability potentially to provide quantitative information and being not so affected by the superficial layer of the head. On the other hand, we have diffuse correlation spectroscopy, which again is an advanced technique similar to laser Doppler, which is a well-established technology in the clinics. And so by this technique, we monitor blood flow in the tissue. The combination of the two provides information on cerebral metabolism. The new things, the main feature of this project is that we were working in a different scenario. So it was the first time in my life that I was not allowed to do any research in this project. So research was banned. So we only had to complete research, take up completed research and development work, and bring this device to prototype, already tested prototype to the level of a demonstrator. So it was like an innovation, not a research project. And starting from, let's say, a TRL, technology readiness level of about 5, we wanted to push this to something around 8. So a device that can be potentially enter into the market for clinical use. How we did that? So we had the first phase in which we defined all the components, the concept. Then we had quite a long phase of integration, building of the system, testing and validation in the lab, and finally operational phase in real-life settings, which lasted till these months. I don't want to spoil any of the results that will be presented later in other presentation, but just to make a few comments. So, okay, we succeeded in building the device. An example is over there. Actually, we built 2 devices. Photonics was able to provide 1 device in Copenhagen and another one in Milan. And the device were operated by the medical doctor with the same minimal support from developers. Then we can discuss about this. Then, from the clinical point of view, we were able to enroll more than 60 neonates in the study. We got some qualitative acceptable results for tissue oxygenation and blood flow index, and very likely we were happy with this reduced variability in the monitoring of both tissue oxygenation and blood flow index. Anyway, more details will be given in the next presentation by Gorm Greisen. And also, several activities were done in the field of exploitation. So, as said, we didn't just want to make a new device, but the device had to survive after the project. So a lot of activity where major steps were done towards industrialization, production, and this nice sentence, so bridging the gap between research, production, and commercialization, which is the key point of this type of project. And again, this will be nicely presented later by Udo Weigels. And also, in this project, I had the experience of a very nice dissemination, communication, and outreach activities thanks to our partner in Fondazione Politecnico di Milano. We had several press release, newsletter, contact with social media, and also with broadcasting and general public events. Life goes on, so from the beginning of the project, we were thinking of what's going to happen after that. So we want to extend the use of this device in the clinics just to complete some studies that will lead to scientific publication, of course, because also we are interested in that. Udo and colleagues in Photonics are completing the pre-market certification to obtain all the CE mark, and of course, the device will enter in the medical device market sooner or later, as Udo will say. As a scientist, I might say that, okay, nothing is everything good, but there are some open issues, and will be also pointed out by Gorm and other discussion in the round table. I might say that, okay, we can have some hardware and software improvements in the device, in the probe. We have to improve the user friendliness, and most of all, we have to improve data analysis and quantitation of the results, because from the qualitative point of view, we are quite good. Quantification might be we need to improve. And the aim is to move the device from the clinical research to the clinical care, which is the best success in the long term. And okay, just as a conclusion or a roadmap to the future, I would like to point out that this project was a nice project because in the same place we gathered many experts from clinical end user, researcher with the new technology, industry. We supply existing and very nice components. I forgot to mention also PicoQuant, Fraunhofer, and Loop, the other companies. And also, as I said, we need also a medical device manufacturer, as Photonics was our expert in this field. And most of all, funding agency, because all this sector must be funded simultaneously in order to get success. So thank you again to Tanya and the European Commission. And finally, I would like to thank you all. I mean, it was a pleasure for me to be coordinator of this project, so thanks to all the partners and also thanks to the parents of the neonates that were enrolled in the study. So, and thank you for listening.